Hello everyone. So we are in the seventh session of bridge course. In the first session we talked about the cell, and in the second session we talked about the classification. In the third session we discussed about the plant kingdom. The groups comes under the plant kingdom. It means we classified about the plant kingdom. And in the fourth session, fourth, fifth, sixth, we only talked about the classification of animal kingdom. The animal kingdom, as I told you, it broadly made of two groups. One is chordata and non-chordata. Under that, number of phylums, subphylums, classes, superclasses, number of all those. And with all those phylums and classes, we have read the examples or the organisms, animals, and also we have come across, and also we learned the small description, a brief description of all animals. So with this we are going to learn an interesting topic today. So I am taking one interesting topic which is very important to all the living organism. That molecule or that concept or that content is very essential, as I told you. So from that the protein synthesizes. So what is that? Can you guess that? Yes, it is none other than DNA. So the DNA, it is very interesting and important content, based on the class 11 and class 12 syllabus. So this concept, it is there in your curriculum in both class 11 and class 12. Because of that I have selected this. I'll give a small introduction and I'll describe the structure of DNA today, so that you'll have a brush up of the content. Okay, so next, so as I told you, I have selected a DNA. So what is DNA? Why it is much essential? Why it is much important? So the DNA, it has genetic material. So the genetic material, this DNA, it has a number of codes. So that codes, it is read by the sequences, nucleotide sequences, the DNA responsible to synthesize mRNA. From the mRNA, the mRNA synthesizes protein. So not only that one, the DNA, whatever the genes present in that, the genes are also known as the inheritance molecule, the inheritant substrate or inheritant part. So with this we have the content known as DNA, which is very important for an organism to survive or to growth or to development. So where will be the DNA? So you are not first standard or second standard kids. You might have studied about the DNA, about the structure and all. So coming to the eukaryotic cell, if we study the eukaryotic cell, the DNA is tightly packed, or it is covered, or it is present in the nucleus. But here I am taking the word DNA, but DNA actually it is tightly compact, or it is present. Let me write the it forms a beautiful structure known as chromosomes. It means, if I draw the chromosome structure here, the chromosome, it consists a double helical model of DNA. It means DNA is tightly packed and form a shape known as a chromosome. So these chromosomes are present in the nucleus. Okay, so at the time of cell division itself, the chromosome comes to an exact shape, other than it will be in a Like I'll draw here the chromosomes or thread-like structure will be present inside the nucleus only at the time of cell division, at the time of metaphase or after prophase and then metaphase. During these stages, metaphase and anaphase and telophase, only during these phases the chromosome we can observe, we can see the exact shape of chromosome, and other than the chromosomes will be a thin long thread like structures. Okay, so what will be there in the chromosome? It will be there, the DNAs will be there, double helical model of DNAs will be there, and in eukaryotes it is well protected by a nuclear membrane or nucleus, but not in prokaryotes. Prokaryotes have a naked nucleoid, or it is having naked DNA. Okay, it means it is not protected by any nuclear membrane and nucleus and all. Okay, so as I told you, DNA is a double helical model. It looks like a twisted ladder, right? So it is important to study the expansion of DNA. So many of you might have understood about the concept DNA, DNA, what is DNA, functions of DNA, how it replicates, how it looks. But let me tell you from the beginning, the DNA, the expansion is deoxyribonucleic acid. Deoxyribonucleic acid it is. The DNA, it is made of number of repeated individual units of monomers known as nucleotides. Yes, my dear students, nucleotides. What it is made of? If we start studying the chemical composition of DNA, how it looks? It looks like a twisted ladder. What is the importance of DNA? DNA includes a genetic material and it synthesizes the, not directly, first it synthesizes the RNA, mRNA, then later on from the mRNA protein synthesis. Okay, and in the gene, it includes the genes. Genes are responsible for the expression of what enzymes or traits or characters, number of contents, number of metabolical and physiological functions takes place from the genes. All those phenomena takes place or start from this twisted ladder known as DNA. So what it is made of, from what it is made of, what is there, what are the chemical composition of DNA? The DNA chemical composition is, it is made of an repeated individual units of monomers known as nucleotides. Then what is nucleotide? Nucleotide is made of three major parts or three parts. One is phosphate group and sugar. Let me write the sugar, not a pentose sugar, and then next is nitrogen bases. All together form a nucleotide. So one nucleotide, one more, number of nucleotide together forms a polynucleotide chain. As I told you, it is a twisted ladder. How the ladder will be? Ladder is like this, right? It has one strand and one more strand connected by number of links. Like that, we also need one strand, two long strand, and for that two long strand we need a bridge or connecting link how these phosphate group sugar nitrogen bases get connected with one another to form a complete helical model of DNA. Yes, it need a covalent bonds. Yes, my dear students, covalent bonds in the form of phosphodiester bonds and hydrogen bonds. So these bonds connect with one nucleotide to one more nucleotide, one strand to one more strand. Together all these chemical compositions and nucleotides and bonds form a long polymer of DNA. So you can see this diagram. I have not drawn like this. Simply an irregular shape I have drawn here. It means this is the long polymer of DNA. At the time of cell division, it comes to an exact shape. Okay, so the thin thread like chromosomes, inside the chromosome what will be there? It will be a double helical model of DNA. Yes, my dear students, the DNA it itself replicates. One DNA strand replicates into two. How? Let us study. Before that, we will understand the structure of DNA. See, the structure of DNA, one nucleotide strand and one more nucleotide strand. Okay, and there are nitrogen bases connected by one another with a hydrogen bonds. So as I told you, a nucleotide is made of three major parts. One is phosphate group and sugar and a nitrogen base. So this is how they get connected with one another. So it has a 5-carbon sugar, deoxy sugar and a nitrogen bases. The nitrogen bases are 4 in number. There are 4 types of nitrogen bases. One is adenine. Though we are talking about the DNA, let me tell you only about the nitrogen bases present in the DNA. So the DNA is made of 4 nitrogen bases. One is adenine, next is guanine, next is cytosine, and the last is thymine. So those 4 nitrogen bases are connected with one more nitrogen base, one nitrogen base in one more nitrogen base. So like this they connected by a hydrogen bonds. Let me tell you, let me write that: adenine and guanine and cytosine, thymine. We used to call this as purines and pyrimidines. The purines connected by hydrogen bonds, 2 and 3 hydrogen bonds, pyrimidines connected by a double or triple hydrogen bonds. So we are talking about the purines and pyrimidines. What are those? Those are the 2 types of nitrogen bases. Purines, it has adenine and guanine, and pyrimidines has cytosine and thymine. Always adenine pairs with the thymine and guanine pairs with the cytosine. So like this they have you can see the bonds which is present in between purine and pyrimidine. There are two hydrogen bonds are present in between adenine and thymine, purine and pyrimidine. These two considered as purine, these two considered as pyrimidine. They both, all those considered as a nitrogen bases. Okay, and guanine linked with the cytosine by forming 3 hydrogen bonds. So as I have drawn here, so this is one nucleotide. Like this, number of nucleotide together they form a DNA, a complete double helical model of DNA. And if we talk here, consider this is only one strand. I'll draw here. Think of this. Okay, so one strand going parallel in a parallel direction, like here we have a nitrogen base and a sugar nitrogen base and phosphate group. One strand is going parallel direction, having the moiety 3' to 5', but one more strand run anti-parallel direction, having the moiety 3' to 5'. Like that, they run anti-parallel condition. Just take this as an introduction clause or a small description to make you understand. I should draw a complete chemical composition of double helical model of DNA now, but here in a simpler way I want a twisted ladder. If you take one ladder, you just twist it, how it looks, in the same way the DNA looks. Then who designed this structure? Who described the structure of DNA? The scientists are Francis Crick and one more fellow who discovered this is James Watson. James Watson and Francis Crick in 1953. They both discovered the double helical model of DNA. They took some X-ray diffractions and they completed their discovery, and for that they shared a Nobel Prize. So we have the structure here. Only I have drawn the strand here. There are nitrogen bases present in between the strands. If it want to take a complete 360-degree turn, it involves what is that? 10 nitrogen bases. In one complete turn of DNA, it will be included 10 nitrogen bases, in sense these nitrogen bases, these bridges are there, no? So those are nothing but nitrogen bases. One nitrogen base, consider this as an adenine. One more nitrogen base, consider this as a thymine. They both connected with one another by forming a double hydrogen bonds. Like this, number of nitrogen bases get connected, they linked with one another and form a twisted ladder or double helical model of DNA. And the hydrogen bonds are, they can dissolve. Why I am talking about this? How they can dissolve? So at the time of replication, the one enzyme, it helps to dissolve the hydrogen bonds, so that when the hydrogen bond get disconnected, the double helical model, it will open. So how we can draw an open double helical model of DNA? Like this, it will get open. Okay, so one strand, one more strand comes and coil with this strand. and one more strand, fragments of nucleotide join to this strand. As I told you, we are going to learn also about the replication, how the DNA replicates, how one DNA divide into two. This is how it need. We have to study about number of enzymes and number of concepts only. Only a brief content I am telling you here. So the enzyme, first one enzyme helps to break down the hydrogen bonds present in between the nitrogen bases. When the strand is free to get coiled with one more strand, the only one strand comes and bind to this strand, to one strand of DNA helical model. One more strand is free here. To that, there will be a complete single strand will come and bind. Only fragments of nucleotide bases or sequences will come and bind. Okay, before binding to this strand, they will join with one another and they'll coil. Now, so these two strands are DNA strands. And then what else? Now we got the two DNA strands. From one DNA we got two DNA strands. Now how the transcription occurs? Transcription is nothing but synthesis of RNA. How RNA synthesizes? RNA means what? RNA is nothing but ribonucleic acid. What is the difference between ribonucleic acid and deoxyribonucleic acid? Here only the difference is the pentose sugar what we have drawn here. First, second, third, fourth, fifth, CH2OH. So what I have numbered here, no. So this is the first carbon, second carbon, third carbon, fourth carbon and fifth carbon. So RNA got ribonucleic acid. They have that nucleic acid will be having the OH group, two functional OH group. But in DNA, it has only one functional group. That's why it is a stable molecule, and because of the absence of one oxygen molecule in the sugar or in this pentose sugar, it is known as deoxyribonucleic acid. The name itself saying deoxy, dissolving of oxygen molecule. So the name itself saying deoxyribonucleic acid. So that oxygen molecule is present in the RNA. Okay, one difference. One more difference is the RNA. In the RNA there are also 4 nitrogen bases, other than the phosphate group and everything will be common, but in the RNA it has only one strand, but in DNA it has 2 strands. Yes, RNA has only one strand, but in DNA we can see 2 strands. And one more is I would like to tell you, the RNA ribonucleic acid, it has 4 nitrogen bases. 4 nitrogen bases. Can you name the DNA's 4 nitrogen bases? Adenine, guanine, thymine and cytosine. In the RNA, this also consists of 4 nitrogen bases: adenine, guanine, cytosine. One more will be present. Instead of thymine, the RNA is having uracil nitrogen base. Yes, thymine is replaced by uracil nitrogen base. Okay, so we have completed. To some extent we have completed the introduction and the function and the replication and about the RNA, but don't consider this as a detailed content. You are going to learn about the DNA and RNA in this year and also in the next year. Based on that, number of questions will arise. You have to answer to all those if they ask in the NEET. And thank you.